Hello there. We need to start chapter 13, and that comes with notes again. So chapter 13 is based on these slides, just like chapter 12, and then after you get done with the slides, we'll then move on to new activities. But let's jump into it. So you need to get to Google site, and we want to click on, there's two sections for chapter 13 notes, and you'd be happy to hear it's about 20 slides. Chapter 12 had many more slides than 20, so this is should be a faster process for you as far as getting through the notes. Let's check it out and see what it looks like. This is a new format. The last time you looked at slides for chapter 12, they were PowerPoint slides. But what I've done is I've transferred the PowerPoint to Google Slides, which means you can literally type in and edit this if you want. You can write your answers in here as you want. You can just click below and you can add your answer as you want, just like you see there, to any questions there. So you don't have to, you could even write your answers down here. You could add notes. So your responses to question 1 could be right there. So you don't have to open up a separate document to type your answers in. You can type it into the notes section lower here, or you can actually type your answers onto the slide itself. And that's what I'll look at for grades too. And that's where I'll look to check for your progress. I'll look for either you writing notes down here as your answers or writing them into the slide itself. Just depends on what's easier for you. All right, chapter 13 is about genetic technology. And the first slide asks you some questions. The first slide says brainstorm. It's where it says brainstorm web. This is called a brainstorm web here. And the idea is to think about genetic technology. So what do you think about, what words or ideas come to mind when you think about genetic technology? And what you do is, wherever you want to write your answers, you think, what are 4? So the bubbles here represent how many ideas you should share about what you think or know about genetic technology. And then you write your answers here, down in the note section, or you write it on the slide. All right, the second question is, recognize a process or concept you'd like to know more about when you think about the future of genetics. So what's something interesting you think we might be able to do with genetics in the future? And then you'd write your response to that. It's an opportunity for me to see what you're interested in or what you think is neat about the future of genetics. All right, the next slide requires a fair amount of work. You need to come up with a scientific problem, and I can help you with that if you need. But anytime you feel, this is a whole assignment here. So slide 2 is its own assignment. And it's based on this video here. So you're supposed to watch this YouTube video. So you click on the YouTube video, and you're watching the video, and you try to say, what kind of question can I ask myself that's important about this video? If you're confused by this task, you can ask me, but let's see just by clicking on it. So by clicking on the link, you can just tap it, and it'll take us there. All right, so this video has something that says restriction enzyme. Now, we don't necessarily know what a restriction enzyme is, but what you can do is just based on the video. So you watch this whole video, right? You just watch it, different things are happening here, and you ask yourself a question that you think is important about the video in order for you to learn more about it. So what question do you need to ask yourself in order to better understand the process from the video? And that's where you write, oh, wrong one. And that's where you write right in here. So what the scientific problem is basically a question you ask yourself to better understand the video. So what should you ask yourself in order to better understand what that video is about? And the title was restriction enzyme. So a possible idea could be, what is a restriction enzyme? That's a fair question you could ask yourself. What are restriction enzymes? And then the next part one thing I've noticed from the Google Slides is they look tiny. You want to make them look larger, right? So the best way to do that might be zooming to fit in. So you can use this zoom function, or you can use this right here. So this really blows it up. It makes it much more zoomed in, more than you might want, but just try your best to work with it. You tap it once, and you see it zoomed in a lot. So what I'm going to do is I'm going to double tap it. That way it shuts it down. It's not going to zoom in anymore. So if you want to turn off the magnifying function, you've got to click it a second time. So you click it once, you come over here, you tap it, and once you got it to the zoom you want, go back, tap it again, and then it's off, and you can use the regular cursor. So now that it's all zoomed in, I can only see one part of a bigger thing, right? So I need to scroll over, and I need to also, let's see here if I can find it. I need to go. So there's the problem. You can type your name in here too. But you have to always erase the line before you type into it, right? And then, so what happens? So you watch the video, and then you write. It's wanting you to write 1, 2, 3, 4, 5, 6 observations from the video. So you watch this video, and you come up with different things you noticed, different things you understand, or what do you see happening. There's no perfect language to use. You may not have perfect academic language to use in your observations. It just has to be your best effort to use the best words you know to document what you're seeing in that video. And that's 6 total observations. And these 6 observations are going to be the basis for you to try to understand more about that question you wrote up top here. So for example, if you asked what are restriction enzymes, well, the video is about restriction enzyme. So you write down all the things you see, and hopefully as you go down further in this chart, you can explain what you think is going on. Right here, this hypothesis section, a hypothesis is your effort to answer the original question. So if your original question was, what are restriction enzymes? Based on what you wrote here as observations, you come here now and answer it. What do you think a restriction enzyme is? What do you think its job is? What do you think it's doing? And then you just type it in there in that spot. And that would be your hypothesis. The final thing you do is really connect everything that you've done. What was your original problem? What are the observations you had? What was your hypothesis? And basically, in your statement and claim section here is you type up, not copying everything that you did above, but you're connecting all those things into kind of a new paragraph statement. And you should be able to write at least 4 sentences about what you've learned about blank. What can you now say about restriction enzymes? Where you can say, well, at first I didn't know what restriction enzymes did, but after taking 7 observations on what they do, I now think its job is, and you could say right here, like, this could be your hypothesis of what a restriction enzyme is. That's when you could share it. You're like, based on my observations, I think a restriction enzyme does blank. I think that because, and then you give reasons why you think what you think, or why you have your hypothesis, and that should be based on your observations up here in this top box. So you can say, well, my hypothesis is that a restriction enzyme is blank, and I think that because of this reason, this thing that I saw, and this other reason. And by the end of it, you should be able to write at least 4 sentences telling another person what you think a restriction enzyme is and why, providing evidence. This is all about connecting what you see and using these things that you see, using your observations as evidence to support your hypothesis. So you have your hypothesis here. What do you think a restriction enzyme is? And then why do you think it, right? And that's what you write right here. So you're connecting your ideas, your claims to evidence. So you're backing up what you say with your observations. That's all done right here. All right, good luck. The next slide, surprisingly enough, here, let me go back to the bigger view, is restriction enzyme. Again, and if it's too small for you to read, then you just head back over here to the zoom in function and you click on it and make it a little bigger. So on this slide, it defines what a restriction enzyme is, which is pretty cool. And so when you read through this, you can compare what you did with slide 2. So slide 2 is your first opportunity to kind of explore what it is, and then compare it to what the textbook says a restriction enzyme does. So this definition is based on the textbook. Your job here is, it says, so we have our definition, we have links also. So you can click on 3 different links that should help you better understand restriction enzymes. So I already watched 1 video, this is 3 more you can watch to kind of understand what restriction enzymes do. What are they doing? Now if you look here, you see A's, G's, T's, C's, A, T, C, and G. Yes, sir, that is DNA. So we're in genetics, and DNA is always a focus. So it says a restriction enzyme breaks nucleotide bonds only between the 2 A nucleotides. Let's zoom in, and let's better see this. I'm going to double click it so the zoom function goes off. Then come over here. Look, it says restriction enzyme breaks between the A's. Here's the A's right here. So if restriction enzymes can break chemical bonds in DNA between A nucleotides, so right here and in between here, there's 2 A nucleotides here. So a restriction enzyme, this little blue thing here, can come over to a DNA strand and cut it in certain sections where 2 A nucleotides are next to one another. So it can break the chemical bonds of DNA and separate parts of DNA. if it can do that, which one of the strands here, A, B, or C, which one of these strands would result if which one of these pieces reflects the restriction enzyme cutting between the A's here? Between A's here, between A's here. What would that piece look like? This, or that? So you choose one. It says, number 2 here says, defend choice with evidence. So why do you think it's A, B, or C? You provide a reason. And then the third question is, what do you think the value is in genetics for restriction enzymes? What could be good about something that can cut sections of DNA out? Why do you think it might be valuable? And so you can read the original definition again. So you can read this definition, or you can just think creatively on your own. What do you think is the value? And you put my hypothesis is. So you can type your answer here, or you can type your answer down below. So for questions 1, 2, and 3, you can write your answers down here in the notes section or within this box. Could get a little messy if it's in the box, though. All right, moving on. Number 4 is another activity, just like we've done already. But we have a new website. So it's the same task. Come up with a question that will help you learn more about something. You come up with your observations. You write your hypothesis. And then you kind of take all that information and kind of put it all together. I think what I think because these are the things that I saw. So you just come up with a hypothesis or a claim, and then you support your hypothesis or claim with the evidence that you found or your observations. And you should write at least 4 sentences. Let's look at the link though. So right here, you can just click on it, it looks like. There we go. So it takes us to that. It says polymerase chain reaction. So you have a video right here. So this time you're learning about polymerase chain reaction. So the original problem, I would say, should be at the very top. I would write, what is polymerase chain reaction? And then watch the video. So you watch it, and you study what's happening. There's a little audio there. Don't worry about it. All right. So you get your observations based on the video, and then you carry on coming up with the full statement. All right, the next slide is us talking specifically about polymerase chain reactions. So it gives a definition, and you can compare that again to the work you did here. So anytime you're doing work here, use it to base on how close was I to understanding what it did. So read this, and if it matches well to what you wrote here, you're doing well, you're learning. All right, let's look at the task. It's easier to study a single gene or the same gene copied a billion times. So what do you think? So the scientists like to make a lot of copies of DNA. Do you think that's helpful in any way? Do you think there could be, why do you think they do that? Why do you think they make so many copies of DNA? And it's just your opportunity to share. Why do you think they do that? And there's a hint here. It says, what's easier to study? A single grain of sugar or looking at a giant pile? So what's easier to see and look and interact with? A single grain of sugar or a giant pile of sugar? And so relate that idea there as a hint to why they might be making so many copies of DNA. And the last thing is, how could PCR be helpful with a small amount of evidence or DNA found at a crime scene? So how could detectives, people studying the crime scene, collecting DNA evidence? How could this PCR help them with just a tiny amount of evidence they found at a crime scene? And again, you write your answers here on the page or in the evidence section. All right, next is showing you more about restriction enzymes. Read through the question. Read through the question again. Let's see this first one. Construct a 3-step strategy that uses restriction enzymes to ID a guilty person among five suspects in a criminal case. So if everyone has unique DNA, everybody has DNA specific to them, and it's not going to be identical to somebody else. And if they can use someone's DNA to identify who's the criminal, how would that look like? If you were trying to create a 3-step strategy that allowed you to choose certain sections of someone's DNA and also allowed you to know what the DNA is at a crime scene, how would you try to create a 3-step strategy to use restriction enzymes to identify DNA of the criminal involved? So, and there's no perfect answer. I'm just looking for creativity. And so you can write your 3-step strategy down here. And then the second question is, Well, we'll have to wait.